In this video, we're going to continue our discussion of microbes in the lab. And first, we're going to start with defining what is a media. So a media is going to be a nutrient source that we're using to grow organisms outside of their normal or natural habitat. Media can be classified based on its physical properties, chemical composition, and even its functional use. So for its physical properties, there's a variety of media available, but the two most common physical forms include the liquid and the solid states. Now, the liquid state means just that. It's going to take whatever shape of the container it is in and can move freely around in that container. In lab, we're usually using flask or we're using test tubes. The liquid state is also referred to as a broth. And if we want to make a solid media, we use that liquid media as a base. We add a substance known as agar, usually about 1.5%. We add that to the broth, we heat it up, and then we pour it into the appropriate container or vessel. We allow it to cool, and then upon cooling, it becomes a solid surface that we can then use for incubating, etc., microbes. Another way we define is by the chemical composition. The chemical composition can be either defined or complex. So if it's defined, that means we know every component that's in that media and that it's 100% composition known. Okay, that's going to generally be a synthetic media that is created in lab. Most of the media that we are going to use is going to be a complex media because there's at least one component that is not chemically defined. Now, why would that be? It's probably because we've added a natural component in there, something from a plant, animal, or even a yeast, something like blood or serum and yeast extract, etc. And so because it's natural, we don't know 100% all that chemical composition, and so we refer to it as a complex media. We also will define medias based on their functionality. Now, when we use a media, just remember that we are not going to examine individual bacterial cells. That's what we use a microscope for. In the case of medias, we're focusing on the growth of that bacteria on the media, and when it's a growth and it's a large number, we refer to it as a colony. When we're just looking at a media and we're looking at the observable growth of the microorganism, we call the whole thing a culture. So when we're looking at medias and we define them based on their functional, we have enriched, selective, and differential medias. Now, the enriched media is just as it sounds. We've enriched the media. We've added something to it. So we've started with our very basic media, which we refer to as a nutrient agar, okay, or nutrient broth, and we take that basic media and we're going to add a substance to it that helps or promotes the growth of a specific bacteria. So in the case here, the example, we've added some blood to a media to enhance the growth of fastidious streptococci. We also will use media known as selective media, and just as the name applies here, in the case of a selective media, the media is going to allow for or select for one type or one group of bacteria. It can be very specific or it can be a little bit more broad, such as for gram-positive or gram-negative. So in the case of selective media, we've taken that basic nutrient agar and we've added one or more substances to it that's going to inhibit the growth of one type or one group of microbes, but then it's going to allow the growth of others, making it easy for us to select for or isolate out the particular bacterium. So for example, in lab, you're going to use a McConkey's agar in which we've added bile salts, and that has then inhibited the growth of gram-positive and allows for gram-negative. We also will use media based on the ability to differentiate between the bacteria that actually grow on it. Okay, so bacteria that grow, there's color changes that usually occur, and we're able to differentiate bacterium, and so those cases we call it a differential media. Just like we did with selective media, we're going to take our basic nutrient agars and we're going to add different chemical or compositions to it that is going to allow the metabolic processes of the microbes when they're growing on that media. Those metabolic processes will then allow for a color change, something that is going to look very different. You're going to have a physiological difference. So as you can see here in this slide, you have the media has changed from yellow to pink, or actually it changed from pink to yellow, but you can see a color change. In some cases, it's the colony itself that changes the color. But in a differential media, it's going to allow us to differentiate or distinguish between the types of bacteria growing on that agar because of color changes in response to metabolic activities. Okay, so if the bacteria is producing an acid or base and things along those lines. Now we're going to shift gears just a little bit and talk about the microscope. The microscope is going to be one of the most used tools in our lab. Notice here that I just have pictured the microscope. You do not have to worry about the parts and the pieces and all of that as much in lecture, but you do have to worry about those in lab. Okay, for lecture we're going to look at a lot of the concepts of why we use a microscope. So why do we use the microscope? We're looking at microscopic organisms, which means that we're not able to see them with the naked eye, so we use that microscope to help us enlarge and make it so that we can see the microbe. Now, magnification and enlargement is huge. However, think about like on your cell phone or computer or something like that, when you take a picture that you want to see and you try to enlarge it in order to see a particular piece of it or to distinguish something. If you enlarge it and it becomes so pixelated that it's no longer clear, then magnification hasn't done us any good. You have to be able to magnify with clarity. Same thing is true for the microscope, okay? But in the case of clarity, we refer to it as resolution. So when we refer to magnification, this is something that should be a known fact or repeat from your prereqs. But in the case of our microscopes, when you talk about magnification of the specimen, remember we're using binoculars, which means it's a two-lens system microscope. So you're going to have to get the total magnification by multiplying the magnification of the oculars, those lenses that you'll look through, and you multiply that by the objective lens. Did you use the 4x, the 10x, 40, etc.? Okay. So we're going to magnify, but we need to have clarity. So for clarity, we refer to it as resolution. And in the case of resolution, what we're looking for is the ability to be able to distinguish between two objects or points. And you got to be able to distinguish one from the other. And we're looking to find what's that minimum or the shortest distance. So in the use of the microscope, really the limiting factor for us when making something larger, it's not the magnification ability, it's the resolution ability. So we refer to that distance that allows us to see, get it larger, but with clarity, as the limit of resolution. And there is a formula to calculate that resolving power that gives you that perfect magnification with resolution, okay? And it's the wavelength, which is divided by the addition of the numerical aperture, which is the ability of a lens to capture light of both the condenser and the objective. Now, if you were in lab and you were looking at the microscope, all those numbers would be provided for you and you would just plug them into the formula. So in lab, you'll learn a little bit more, as I said, about the microscope, but the last major point that we want to discuss for lecture is the type of microscope that we're using in lab, and it's known as a light microscope or a bright field microscope. And the reason for that is because we take those slides that we have prepared and we put them on the stage, and the light passes up through the specimen. So in a bright field or a light microscope, the specimen is being surrounded as that light gets moved up through it. We don't always stain it, but we do stain it to help make it a little bit denser and easier to view. But we can also use stained, unstained, live, or preserved specimens, and we use a bright field or a light binocular microscope in our lab settings. So that's it for the microbes in the labs, and look for other videos.